Any questions, coding problems, anything that's unclear? If you want to get ahead of things a little bit, I can put in the chat a link where you can download the things that we're going to be working with today. In the supporting materials for students, there's a document called the Tellurium cheat sheet. I would recommend downloading that. And if you have a printer, I would actually print it out so you have it on your desk. But if not, just have it as a PDF available to you. Download the demos today. You can get all of the demos from here. And the one that you need the most is going to be called Tellurium v2.zip. There are a bunch of, they're all labeled exercise 5 underscore something. I didn't renumber them last year. The module order was slightly different, and so the numbers, it's going to be 5. We're going to, for us, it's going to be 5 underscore 3. And in this, it's going to be 5 underscore 2 or 5 underscore 4, but they're the same demos. And that if you want to go directly to that, just that one Tellurium v2 file, this will take you there. So I would recommend doing the downloads for that.
Yeah, I just want to ask, like, because we learned how to use the diffusion constant yesterday, and then if I want to model a facilitated diffusion or active transport, um, um, how should I do that? Do I edit it every step or?
What kind of active transport?
Actually, I want to model a GLUT1 glucose transport that is a facilitated diffusion. So it depends on how many GLUT1 is there
You
on the
mean it's on the membrane?
yeah, yeah, yeah.
So there is a new diffusion solver. The diffusion solver FVM supposedly lets you do this. Typically, I would do this by hand.
Oh,
So I would model the. I mean, the one thing that we do not have in CompuCell at the moment, and
mm-hmm,
none of the packages have. Uh, the one package that has this in detail is VCell from the University of Connecticut, from Blinoff at University of Connecticut. That has very detailed model of ion channels and transporters.
mm-hmm.
But it historically was only for modeling a few cells, and didn't have cell movement. It would be very interesting to combine the two things together. Depends a little bit on how much detail you want. But
Mm-hmm.
if you have a model with a field inside of the cell, and you can, it depends how many cells you want to model. If you're only going to model one cell,
Yeah.
in CompuCell, you could build a single cell. You could build a layer of another material, which represents the membrane, on the
Mm-hmm.
surface of the cell. And then you can actually put little subcells which represent ion channels in and have different transport rules. And in some years, we've had more time and we've actually done some modeling of that. But it's
Oh,
pretty expensive, so you can only model a couple of cells at a time when you do that.
I see.
Now, if you want to model a lot of cells, then at the moment, what you would do is you would do exchange by hand, basically, where you have a flux. You look at the intracellular concentration, you look at the concentration of ion channels, and then you secrete and absorb based on that.
Oh,
What's a bit of a problem is that we can have the ability to model effectively secretion over the whole cell surface,
mm-hmm.
but you can't have different secretion in a simple way with a single cell. Um, you can't do secretion to local extracellular environment, which is different at different positions on the cell. And so it depends a little bit on what kind of situation you're in. If you
Mm-hmm.
want to do lots of different regions that have different secretion rates,
Mm-hmm.
then you could do that. You use the compartmental cells that you're gonna talk about next week. That if you heard the talk by Shane Hudson yesterday, he
Yeah.
showed how to do compartmental cells. So if you do compartmental cells, then you could have compartments on the surface of the cell.
Mm-hmm.
Each compartment could separately have a concentration of ion channels, and then you could do secretion and absorption from each compartment separately at the rate you want based on the transport rules locally to the field at that point. Again, when you do those kinds of things, it's more expensive, so you can't model as many cells at once. On the other
Mm-hmm.
hand, if you really care about the spatial variability around the boundary of the cell, then you have to have a lot of spatial detail. So
I
the answer is you can do it, but it's work. It's also pretty straightforward. It's not, I would say, it's relatively straightforward to do it in two dimensions
oh
in a section. If you really want to do 3D cells,
yeah.
then it's more expensive because you have a lot more compartments you have to look at. And this
Yeah.
happens also in 3D. I mean, it's not something specific to CompuCell. This is a fundamental issue with
Mm-hmm.
this kind of model. If you used a finite element model, you'd face the same things.
Yeah.
So it really comes back to how much detail you need
I
for
see.
the particular biology.
Mm-hmm. I was thinking about just the red blood cells, so
I'm
I
sorry,
think
what kind of cells?
red blood cells.
Oh, red blood cells.
Mm-hmm.
So you're trying to model oxygen transport then, or
Uh, the glucose transport and the
glucose?
effect on the cell shape.
Ah, so you
Yeah.
want to do okay.
Yeah.
So so yeah, that's rather a different set of questions.
Mm.
The If I think about glucose, I mean about red blood cell modeling, there's been a lot of very detailed biomechanical red blood cell modeling by Roger Cam at MIT, George Karniadakis at Brown, and others. That's very heavy modeling, where they do finite element mechanical models of the red blood cells in great detail.
Mm-hmm.
Um, and that's sort of a specialized technique. I mean, those are techniques that are essentially used only for modeling red blood cells in blood. Um,
Oh.
haven't been used in much other, and I don't know if their software is available publicly.
Mm-hmm.
Uh, but
Okay.
it's beautiful work. Um,
Mm-hmm. Yeah.
so again, I'd be happy to talk to you more
Oh,
about that and try to
yeah.
understand. But in general, you know, oxygen and glucose have such high diffusion coefficients
Yeah.
that you don't get a lot of spatial variability over the surface of a cell. Especially red blood cells aren't that big,
Yeah.
and so the oxygen and glucose variability in the environment of the cell isn't that big.
Uh-huh.
And so you don't need a lot of spatial detail in the diffusion fields for that.
I see. I see.
Um,
Yeah.
I mean, I can imagine situations in which you do need it, but we did that little small liver, that two-dimensional liver model, which was
Mm-hmm.
very simplified, but we did actually model in a very simple way fluid flow through a capillary. The capillary carried
Yeah.
simplified red blood cells, and
Mm-hmm.
those red blood cells did exchange chemicals with in that case, in the liver, the hepatocytes. And we could see exchange of chemical between the cells of the hepatocytes. Um,
I
so you can do it with CompuCell. We've done that kind of thing, but there wasn't a lot of. We weren't modeling the shape changes of the red blood
I
cells
see.
in that
Mm-hmm.
in that situation.
I
Um,
see.
the detailed mechanics of red blood cell reorganization are pretty
Yeah.
complicated, as you know.
Yeah.
Um,
Yeah.
if I wanted to really model the
Mhm.
red blood cell shape in detail,
Mhm.
I would probably use a finite element approach. There's a package that we started developing in my lab that's now developed out of the University of Florida called
Mhm.
Tissue Forge, which is a very high performance
Mhm.
particle-based modeling method that can be used to do finite element as well. And I would say that probably that would be the best package to
Mhm.
develop this kind of, if you really want to do the fluid, explicit
Mhm.
fluid and the red blood cell shape, I
Mhm.
would say that would be the package to use for that.
I
But
see.
I don't know what your level of computing experience is.
Yeah, I'm only an amateur, I would
Yeah.
say. Yeah, my
So
major.
yeah. Sorry, go ahead.
Yeah. So yeah, I actually built something very simple before using like CompuCell 3D. So
Yeah.
if you have time, maybe
Sure.
like yes, like not now because like I see many people have questions,
Right.
but later on, can I
Hmm?
ask for guidance and stuff like that?
Sure. I'd love
Yeah,
to. I mean,
thank you.
I don't want to be discouraging. I mean, the things you're
Yeah.
talking about the
Uh-huh.
the code, for example, that Roger Cam wrote or Karniadakis wrote
Yeah.
is probably 100,000 lines of C++ plus plus to
Yeah.
do just to do simulations of red blood cells in fluid.
Mm-hmm.
So building detailed simulations of the mechanics of red blood cells is quite a difficult problem.
Yeah.
Although people have done it, so there's a lot to use to do it. I would say the Tissue Forge package probably
Okay.
would be the best one for that specific question. If you want in 3D the shape changes of the surface of a red blood cell based on the mechanical properties of the membrane and the actin cortex,
I see.
um,
Okay. I'll
then
take a look into those.
but that may or may not be what you need. In other words, I always say the same thing, which is do the simplest model that will answer the question you're
Uh-huh.
interested in. And so until I know
Yeah.
the details of the question, I can't really advise you on the details. But I'd be
Yeah.
happy to meet with you one-on-one to
Okay.
talk about that.
That's cool.
Anybody, okay, anybody else have any questions? I see there's been some discussion about how to do um, network models, which is what we're going to be talking about in 10 minutes. Um, Are there any other questions or comments or concerns, debugging we can do in the 10 minutes before we start the workshop prompt? I want to say for today, um, there's a document in the student materials folder called, it's in the reference materials called the Tellurium cheat sheet. And I would recommend downloading that to your computer. And if you have a printer, I would print it out and have a copy on your desktop because it's convenient to have it on a piece of paper so you don't have to keep thumbing back and forth between it and your computer while you're using it.
Hi. So CompuCell3D has been opening for me all summer until
Mm-hmm.
today. I just started trying to catch up and run some of the exercises on diffusion in my player when I try to open in player so I can visualize the field. It's just nothing is happening.
What do you mean by nothing? Does the Python console show up?
Um, the player itself isn't opening. I mean, I do see the
But
Python console,
all right,
but
so what error does the Python console show when player fails to open?
so I see before I try opening player, it says import error numpy core multi-array failed to import. I don't know if that would be causing the problem or not. So
And it stops after that, or is that a warning? I don't know.
it's a warning. And
And if it's a warning, then it's not that.
okay. Basically the last message I get is starting CC3D.
Hmm.
And that's
Are you using a Conda install or did you do a one button install on your computer?
definitely didn't do Conda, so must have been
Okay.
a button.
Okay. So I don't know. I don't know what would have happened.
I know my students were
My
having the same problem where their player wouldn't open for some of the exercises I was trying to have them do. So maybe I just need to reinstall.
That's possible. I don't know why. I mean, we didn't change anything. So it's possible there was an update from Microsoft or something else that caused an issue. That's why we went to the Conda installs, although they have their problem. The only thing I can say, and if is anybody else having this issue, I mean, my CompuCell's opening fine today, so I just checked everything. And I wish you'd reported it yesterday because we could have worked on it overnight. But do you want a screen? Maybe somebody could go into a breakout room and try to help out with that. Lorenzo, possibly, could you help?
Yeah, I can.
Because I think I mean, before we do that, is there anybody else who has an issue with this? My guess is that it's probably an SQLite error. So do you remember I said to try debug? Did you try
Yeah.
deleting the
I
SQLite
haven't tried
files?
deleting that yet, so I could try that. I'll just try that.
And you're launching? Is it Twit will launch and Player won't launch, or neither will
Yep,
launch?
it's what it will launch. Just fine.
Okay. And
It's
are
just
you
the Player.
launching Player from Twit, or are you launching it directly?
From Twedit.
Try what happens if you just say CompuCell3D and launched Player by itself. Because if you have if you have an illegal file that you loaded before in Player, and when you launch it from Twit, then it may crash. So
Okay.
I would try that. But why don't we
Okay.
go? Why don't we have before? Let me just give you some other links beforehand. So we're going to be doing some exercises today that will need you to do some downloading. Those exercises will be all under the title either exercise 5 underscore something or Tellurium 2 V2. And if you finding the Tellurium V2 in that folder difficult, I will give you a link that will take you directly to the Tellurium V2 file. And I'd ask you to do the download of that Tellurium V2 zip right now, so you have it in hand, so we don't lose time later, okay? Oh, that's a possibility. So somebody deleted the CompuCell command line, so that's possible. If anybody else is having issues where CompuCell Player is not launching from Twit, again, I remind you about that fix of deleting the SQLite file. I realize sometimes it's hard to figure out which SQLite file to delete because it's a hidden file. We can maybe do that. So is anybody else having problems with Player not launching at the moment? Anybody else need help with Player not launching properly? It's the first time I've heard that particular. We've had Player crash, but fail to launch is not something I've seen.
Yep,
Somebody says they have to launch Player twice. That's interesting. I think I've seen that myself. Is that launching Player from Twit or launching it directly when you type CompuCell3D from Twit? Modcheck never launches Player from Twit. He's always worried about that. I happen to find it convenient to go back and forth. Oh, so one thing that I did notice yesterday, which is if you close Player and then want to reopen it, if you look at the icon in the upper left-hand corner of Twit, there's a little CompuCell icon. If that is gray, the old instance of CompuCell is still there, and you have to wait till it turns blue before you can launch it. So if you click on it immediately and it's gray, it won't launch. So you have to wait. So that's an issue because of the way CompuCell is installed. When you close it, it doesn't disappear right away. That seems to be true of a lot of software now. You hit close or stop, it doesn't immediately disappear. It takes some time to clear it from the computer's memory. So you have to wait for that. If that icon stays gray, Player hasn't closed. So you may have to go into your job control and kill that rogue process direct. And I've noticed that never used to happen, but I've noticed that now for a lot of things. Even Microsoft Word will do that to me sometimes. Word or PowerPoint will crash and then they won't go away. You have to go into the command and close them. All right, that's a great point, Ava. Thank you.
So welcome everybody to day 5 of the CompuCell workshop. This is the last day of our first week of training. Just a reminder that this session, like all other workshop sessions, is being recorded and live streamed to YouTube for future reference. Today's slide deck can be found in the student materials folder as usual. And then we still have our hackathon teaming document available for people to start putting ideas into. I think there is one hackathon slot still available for lightning talks this morning if you would like to try to do your talk today. And if you have not joined the Slack yet, please do so over the weekend. We'll be using that a little bit more next week as we prepare for the hackathon. Just want to say thank you to our funding sources through the NSF and previously through the NIH, as well as our support networks through the IUB Biocomplexity Institute, as well as the OpenVT and Glimprint initiatives. So today we'll be hearing primarily from Dr. Glazier. He'll be doing his 2 lectures on network modeling today, and then we'll have another round of guest lectures in our second module. So again, from 10 a.m. to 11:30, we'll have our first module of the day. From 11:30 to noon, we'll be doing our hackathon lightning talks. Those of you who are presenting, please have your talk pulled up and ready to go because we'll need to jump kind of straight into that before lunch. Everybody will have 3 to 5 minutes to present their talk. Lunch break from 12 to 1, module 2 from 1 to 2:30, 30-minute afternoon break, module 5.3 from 3 to 4:30, and then Q&A with instructors. The guest sessions today will work the same way that they did yesterday, where when you come back from lunch, you will join one of the 3 rooms that were sent by email this morning, and I will send a reminder email kind of halfway through the lunch break today with the links directly to each room. If you'd like to see Tom Knudsen's presentation, you will actually stay in the main session. They'll be presenting here. Toru Matsura, one of our collaborators from Japan, and Jim Sluka, one of the group members at IU here, will be talking today about some work that they published last year. And then Bharat Vivan, who is a previous CompuCell workshop attendee. He attended in 2022, and he has been using CompuCell ever since and has recently published a paper. So he's going to walk through actually his whole process of sort of learning how to build his model and what he had to kind of think about as he went as a new sort of semi-experienced modeler. So you'll join that room and then you'll come back at 3 o'clock p.m. Eastern Daylight Time for our final session today.
I recommend going to the student materials, reference materials, and downloading and printing the Tellurium cheat sheet if you have a can. Because you'll be referencing that a little bit today. And then we'll definitely, for this module, need this Tellurium v2 zip. You'll need it. So please download that. And the other demos are available from the other link that I provided in the chat. And if you can download those now, we'll save you time and make things a little bit easier.